impressed with this sediment here.
Yeah.
Jake, you want to look at the core there, please?
Yeah.
This might even be shallower.
Yeah. That's going to hold.
Is that good enough for you to keep?
No.
No.
I don't think so.
Roger that. I'll toss it out.
Okay.
You want to try a different spot a little further up slope there?
Well, we were going to collect one of the sea cucumbers. Are we close enough to do that here, or do we need to move a little?
Sure thing, yeah.
Well, while the push goes in hand, it's easy to just bump up slope, and there'll be sea cucumbers a little bit further up.
Okay.
So if you want to really get the sediments.
Sure. Let's move a little and give it another try.
Roger that.
Palm trees swaying in the breeze.
All right, we can try here. And if this doesn't work, we can at least grab a cucumber.
Good plan.
Roger that. Raj.
There you go.
Oh.
Going down a bit.
A little bit.
It's pretty good.
Looking good.
Didn't get any more out of it. Nope, that is all she will give me. Apologies. All right, Jake, you want to set up on the crates there?
Yeah.
Get it back before it falls out. Nice.
Very nice.
Good job.
Okay,
Awesome.
so we got a sample.
That's going to be NA124-018 in push core number 11.
I'm going to straighten that out. It's not in there very well.
Tappy tap.
That was orange.
All right, and then a sea cucumber, correct?
Yes, please.
All right, Would you guys want this as a grab or as a slurp?
Slurp, please. And if I think we could maybe fit 2
2?
in the container.
Sure thing. .430 Raj. All right, go ahead and give it a good flush there, Jake.
I hear that, flushing. I missed that. Was that core successful?
It was
Roger.
very shallow, but successful.
11.
Could we get a nice close-up of the sea cucumber before we slurp it, please?
Yes, definitely. All right, Jake, let's call that a good and flush there. I'll move this guy out of the way. Jake, you want to keep an eye on the manip there while we zoom in?
Watching.
Roger that. This is full side that we have there, science. That's not the prettiest of shots, but
That'll work. So here you can see the sea cucumber is very transparent. The head end is looking down towards us with all of those tentacles coming out, and these have sticky little pads on the front, which they use to pick up the detritus to eat. And towards the back end of the sea cucumber, you can see its gut, and it's full of sediment.
Yeah.
What you often see with these sea cucumbers, and you can see with this one, is in the bend of it, there's a purple commensal scale worm. And if we're lucky, we'll bring that scale worm up. These are a well-known group of scale worms that are this beautiful purple that live in association with sea cucumbers all around the world. And also just behind the front end of the sea cucumber, you can see some white internal organs, and that's part of its gonads. So this is very helpful later on for us when we're trying to do any taxonomy on these, because the sea cucumber, unfortunately, as it comes up, will probably really suffer from the loss of pressure and temperature changes. So they often can be pretty in bad shape by the time we get to have them on deck. Okay, that's great.
great.
If you
Oh,
could
I
try and
found
get
it.
the scale worm too, Jessica, that would be really great. Did you see the purple worm on the sediment?
Yeah,
Yeah.
yeah. We'll zoom in too while we go ahead and take a slurp of it.
It should come with the they can swim a little bit, but it should come when you slurp the sea cucumber.
Roger that.
Just confirming which slurper containers is going into.
Great question. That is a good question for you
We
guys.
just did
Number 7 course.
6, so yes, 7 would be
Beautiful.
good, I think.
Thank you.
You happy with that zoom?
Go ahead and do a little bit more there, please. That is wonderful there, Jonathan. Thank you. Maybe I won't let him on that we're going to take him. Get too close. All right, you ready there?
Ready.
Are we lined up on it there? Gonna try jogging forward and backwards there.
Yeah.
Okay, seems like it then.
Yeah, that's the best I can do.
Okay, that's great. Number 7. Go ahead.
Coming up, 50%. Hungry, hungry hippo.
The worm is not there anymore either.
Looks like you got it.
Awesome. They're
Good.
both on
All right,
its way.
full wide there, Jonathan,
far.
please.
Ooh,
There he is.
And there's a worm, I think, coming in as well.
And
Yeah,
if there's another scale worm, another sea cucumber you can get,
Sure thing. Go ahead and zero that one a bit there, Jake.
This is going to be sample 019.
Roger that, 019.
Which so far is
This one's a little out of the view here, but we can go for that one. Or we can hop up and go. It might be easier so that we get a good view of it. There's one on my porch.
Yeah,
What
that
the
was
heck?
the last time you landed. It rolled up on there. I saw it. I saw it in the bubble cam.
Roger that. Well, we can do some self-maintenance if we like. Just scoot over. Any preference here?
Your choice.
All right, did you hear that?
Ooh. Lift off.
Get out of our little dust cloud a bit.
This one's just begging.
Yeah, that one's just asking to be slurped. I don't know if we want one that's... Let's go for this guy back here. Did you want another zoom on this? Or
Yeah, just to see if it has one of those worms would be handy. Thank you.
All right, go ahead. You can actually see the scale worm in the flush jar there.
Yeah.
Oh, very nice. Yeah, that's great.
gorgeous. It's amazing how translucent they are.
Many shallow water sea cucumbers are just basically like sausages, but in many of the deep sea ones, they've evolved these extensions of the body wall that allow them to walk along so they can move farther.
Awesome.
All right, Jonathan, full wide, please. All right, go ahead and push in a bit.
And this is the Panichia, correct?
Yeah. P-A-N-N-Y-C-H-I-A.
All right, Jake,
Ready
go
for
ahead.
section? All right, going up.
Roger that. That is so cool, the way these get suctioned up. Oh my gosh.
Shrimps like, yeah, what happened to George?
Got it.
There it is.
Go ahead and zero that puppy out.
Okay.
Full wide there, Jonathan.
Very good. Thank you.
Sure thing.
Greg, what type of scale worm was that?
Well, I can spell you the genus if you want.
Okay, that would be great. Thank you.
Macelliphala.
Great, thank you.
Macelliphala.
I see why you spelled it.
Yeah. Yeah. They're commonly called Violacea because of their purple color, but that's a North Atlantic one.
Yeah, let's do it.
Bridge nav.
All right, let's see if we can find some rock.
Or some sediment that's a little deeper, huh?
50 meters, 225.
Jake, if you wouldn't mind lining back up on the flush and then putting on the starboard bow there, please.
Have to adjust that. It's a little lower than it was.
Nav, can you remind us how far we are along our whole transect here? Are we about 50% through?
Roger, let me just double check on that. Right now we are approximately 225 meters away from point J. We are behind it along the transect. And it looks like, as far as along from the beginning, we have traveled about 700 meters, and until point L, it's about 650 to go. So we're just about halfway.
Point O is the final stop.
Point L, yeah.
Okay, so we've got about 2 hours to cover the second half of the dive. Should be some cool rocks at the end of it, so just keeping timing in mind. Thank you.
Copy that.
Thanks, Kira, for the reminder. We'll try and get there.
Ooh, look at that rock.
That rock is covered in some creatures.
Might be a little big.
Way too big.
Need the Herc roof rack. See if we can get Thule to sponsor us.
Jonathan, go ahead and zoom in, please. That's great.
Wow. Is that a starfish or?
Yeah, very large slime star, it looks like.
Looks like a loaf of bread.
I do not want to eat that type of bread.
So very full of, pumped up with fluid, with water. So they have a fluid system, hydraulic system inside them, and this one's very inflated.
Is that a defensive mechanism?
Slime is a defense. I'm not sure why these ones do this.
Full wide, Jonathan, please.
Full wide.
I wonder if slime stars have the same type of silky slime that hagfish secrete.
Well, hagfish have these strange proteins that are all folded up, and the moment they hit water, they expand and absorb a whole lot of water. I'm not sure what the slime star method is. They're not as... they can produce a lot of slime, but they're nothing like what a hagfish can do in making a whole bucket of water turn to basically a solid with its incredible protein strands. So in this field, we see a lot of the sea cucumbers. We've got some white snails called Bathybambics. We've got a lot more of those purple sea urchins, or pink sea urchins, fragilis.
And then we're just passing it now, but these rockfish that we're seeing are short spine thorny head, Vastes aliscanus.
And the things coming up that look like windmills are soft coral or a sea pen called umbulella, which has a series of polyps at the end, the dark brown. If you zoomed in on those, they'd all have 8 tentacles.
Oh. Good to go another step.
Sure thing.
Bridge nav.
I'm somewhat surprised you don't see more skeletons of all of
Add
these
one more
sea
step,
urchins.
50 meters, 225.
There might be traces in the sediments, but these sea urchins have very thin shells, very tests they're called. And once they die, they probably disassociate pretty quickly. But if you did a core, there might be some of their spines would show up.
Mm.
So we've just gone through 750 meters. The oxygen level continues to decline. And it will decline for the next few hundred meters. And then if we were to ascend to the surface, when we ascend to the surface, the oxygen level would rise again. So we're passing along or through the oxygen minimum zone now, which in this part of California will probably hit at about 500 meters.
Just as a time check, if we were to go this pace without stopping until point L, it would be just over an hour. So that gives you a sense of how much time.
Okay, good to know. Thank you.
Yep.
And we've got our pull time is at 4 or 4:30?
We'll be on deck at 4:30.
On deck at 4:30.
So depending on how shallow we are at the time, we'll back calculate that with
Okay.
our rate of ascent. But maybe something like 40 minutes plus a little time in addition. So
Should we start another transect now?
Yeah, also wouldn't be bad to, I don't know if Kira's online, if there are any rocks that look particularly interesting to you that you haven't seen before that you would like us to collect, just let us know. But yeah, Transect sounds good, so we can cover
Yeah,
some
we
ground.
need
We have some diverse fauna and geology right here in this screen grab.
yeah, quite a bit.
All right. Transect time.
Yeah, is that officially Transect?
yes, let's start a transect,
Roger
please.
that. Whenever you're comfortable, we'll just keep going at this speed.
Roger that.
Just let me know and I will.
225?
225.
225.
Roger.
So are you going to assign the Transect a sample number as you've been doing? As
Yes,
was
we
the-
will.
All right, stepping forward.
Roger that. So start a transect, and you've been going 100 meters with these transects, is that correct?
Yes, please.
Yeah, this is Transect 4, and we've been going 100 meters.
Roger that.
Transect number 4.
And you had a
Gonna
sample as 20?
Yes, sample's going to be 020.
Okay, I'll mark that at the end of the transect.
Fantastic.
To stop and get them, because I'm a little bit worried about the dive ending and us coming back with ND20 inserts.
We've always, yeah, it seems like we've always gotten to rocky patch. We haven't had.
Hey, Lounge, your mic is hot.
You left the mic on.
Jake's favorite siphonophore,
Yes,
the one that looks
the one
like
that looks exactly like fishing line.
We're all a little shell shocked still, aren't we?
Mhm.
Yes.
Bridge nav.
One sample, I wrote that.
You can add another step to that, 100 meters, 100 meters, bearing 225. Does look like it gets a bit steeper here, so we'll
Okay.
watch our rate of ascent.
Roger that.
So we're doing a transect here of about 100 meters to get us more in elevation, but we're sticking at a certain
Bridge net.
depth and zoom so that the distribution of the animals can be counted accurately, and we'll be doing this on every dive, a series of 4 or 5 transects as we go up each time, so we can compare the different depths and the different regions that we're studying in
Thanks,
the course
Ernie.
of this cruise.
We're okay for now.
Roger.
And if I need to pause, I can pause.
Roger that.
Yep.
Looks like we might be heading out of push core territory.
Yeah. Yeah, maybe we'll find another spot when we get to the top.
That's the biggest rockfish we've seen.
Quite a big one.
Jonk.
Those some of those feather duster worms that we're seeing.
Yeah, all the bright orange are, it's quite a famous worm, this thing called Myxicula. It has very giant nerve fibers, so physiologists back in the days when it was tricky to get good measurements, the model organisms they used were in squids, but also in this particular worm was well known for doing physiology on. And most feather duster worms have a sandy tube, but if we get a chance to look at one of these closely, you'll see that it's a completely transparent mucus. Some of them, as we are flying over, will retract when they detect us, but most of them are staying out of this. It's this distinct orange little circle. I've tried to catch these several times, and they're notorious for having a very long tube going into the sediment. They're pretty hard to catch, so I don't have great hopes that we'll end up catching any of these.
Do they ever bore into the rocks, or are they mostly sediment?
Some feather duster worms can bore into rock. They can secrete acid, and so they go into calcium carbonate substrates, so they bore into coral or other kind of carbonates. This particular one's probably going into sediment.
Very cool.
But we're seeing a lot of them.
Yeah, they're speckled throughout.
So Kyla, those orange worms are called Myxicula, M-Y-X-I-C-O-L-A.
Sorry, M-Y-X-O.
M-Y-X-I-C-O-L-A. OLA.
Thank you. That's cool to learn that they're studied for their long axons, maybe?
Very thick, yeah, very big axons.
I always knew that the colossal squid was studied for those, but
And sea hares,
really
right?
interesting to learn these are too.
Well, you can understand why, and their heads are exposed in a tasty little morsel, so they've got to have a very rapid reaction time when a fish comes along to bite their crown off. So their reflexes are phenomenal, and they have the nervous system to do that.
Wow.
We're getting more and more rocks. As we head up the slope here, we've really left the sediment. But it's pretty heavily dusted with sediment here. Coming up in the frame now is a very strange kind of sea cucumber that glues onto rocks with armor plates and then has a feathery extensions coming out. So it's a filter feeder rather than a deposit feeder, which most sea cucumbers are.
Those branches are spectacular.
Yeah, hopefully we'll settle down and see one up close.
Do they feed during all times of the day, or?
I don't know, Caitlin, about that. There's probably no reason for them not to feed except when they get disturbed and they'll pull their tentacles in. Sometimes we've seen them even today completely retracted and we haven't seen the tentacles and they just look like this armor-plated sponge.
Ah, very cool.
We have about 25 meters left on the transect.
Any idea what these little brown blobs are?
Yeah, they're the worm I was just talking about. I was calling it orange, but you're right, it is a bit browny orange. They're feather duster worm that are going down into the sediment when we disturb them, but otherwise if we looked up close it'd be this incredibly feathery appendages for filtering water.
Awesome.
It's getting rockier and rockier.
Looks like we're going up much faster now, huh?
Yeah, it's getting a little steeper.
Yep.
A little rockier, a little steeper.
So once we've done this transect, I think we'll try and find some rocks to pick up.
Sure thing.
Cool pitting in that rock to the lower right.
It's a beautiful large sea star here. That's actually another kind of filter feeder. This is called a bracingid, just going to the bottom of the frame now. Did you get that, Kyla? Bracingid?
Brisingid.
Brisingid, B-R-I-S-I-N-G-I-D. That's just the family name. I don't know what kind it was. There's another one on the rock there on the right.
Wow.
Man, I've been spelling that B-R-Y for years. There's a mushroom coral.
Also looks like it's feeding.
Oh, big rocks here. Look at that. Another
Yeah.
different mushroom coral.
Could be tricky to sample. Well, there's some small ones, but yeah.
Look at that.
Some more Brisingids.
Ooh. Yep.
Kind of sitting on those rock faces. Here comes some more.
All right, we can mark that the end of Transect there.
Roger that.
And do you want me to hold the ship up here for us to get a rock?
Yeah, I think we will. I think we could settle down past this big rock, and
Okay.
there look to be some rocks we could pick up.
Bridge nav.
Very large sponge there in the.
Ooh, on that rock.
Oh,
Yeah,
yeah.
we might have a look at that.
Hold position.
Sure thing.
Thank you.
And just
Wow.
confirming that was the end of NA124-020 transect number 4.
Roger that.
This is a beauty. Yeah. It's
Look
like a
at that.
shield.
That's huge.
We'll have to see if it's a glass sponge or a demo sponge. I think it's a.
All right, video, go ahead.
You want some more zoom?
Go ahead.
What do you think, Paul?
I think we want to try to sample this.
Yeah.
Absolutely.
Bridge nav.
A little wide there, video. Try to get it full in frame there.
Perfectly positioned at the end of the transect
Do
for
10
us.
meters, 045.
Do you think the manipulator pull off a piece, or I don't know if it would slurp very easily?
We can take a piece of
What
the
do you
slurp
think?
and then, or take a piece of the manipulator and then slurp it in the slurp chamber.
Oh yeah, good idea. Good idea.
All right, yeah, full wide there, Jonathan. Let's see if we can, oh, he's pretty far up there on this little rocky ledge, isn't he? We'll drive into the rock here.
A couple little steps. Yeah, look at that.
That's the biggest sponge we've seen today.
Here's some of the biggest rocks.
Huh. Correlation.
I just backed off 10 meters to arrest that pendulum and get us away from the wall.
Roger that. Thanks, Ernie.
Yeah.
That's a beautiful specimen. Facing the current.
You guys mind if I tried to zoom on Argus to see if we can get a shot of this?
Oh, yeah, sure. Go ahead. Hey, Ernie, you want to press stick lock for me?
Roger. Ready?
Yeah, go ahead.
Stick lock.
We'll see if that settles out. We'll get a better shot of it here. Jonathan, go ahead and push in on Herc HD.
That's a cool shot from Argus
You like that?
and from Hercules.
Yeah, that is.
Hercules.
That's great there, Jonathan.
Roger.
Science, did you want to close up on any of the spongy bits there?
Yes, please. The more close-up we can get, it might help later to identify it.
Roger that.
It's
All
really
right, Jonathan, go ahead and push on a bit more there, please. Take off the scaling lasers too.
Getting some good pictures there, Kylie.
Yeah, I'm grabbing them.
Fantastic.
It's quite nice.
The detail is just absolutely amazing.
All right, there, guys. If you got some pictures, we'll go ahead and come full wide. Full wide, Jonathan. All right. Coming up. Make sure I'm steady here.
Yeah, okay.
Come wide on Argus, please, Jonathan.
How's
I
that?
want to make sure I don't tug you.
All right, that should be all right. All right, and then you want to push in on Herc HD, please, Jonathan?
Zoom, zoom.
That's great there. Thank you. I'm really bouncing around.
Yeah, it might be me tugging you. It is. Do you want me to bump forward or can Jake come down?
Yeah, do you want to do one ship move so I can get a little leash there?
Roger that.
Hello.
Photobombing.
Bridge nav.
Jake, you want to come down on your delta, please, a bit? Thank you.
Do 10 meters, 225.
It's very soft.
It's not a glass sponge.
Wow. It's like that crepe.
Flexible.
Oh, yeah.
Nice.
Nicely
Kind of
done.
like insulation.
Wow.
Fantastic.
Yeah.
Yeah, really cool piece.
So we don't know how old these sponges are, and we certainly didn't want to take a lot of it. So
Jonathan, you want to push in on this, please?
this gives us
That's great.
plenty of tissue for Paul's lab to see if there's any bioactive compounds, and a piece that we can put in the museum for taxonomy.
All right. Got our little quesadilla, full wide, please.
Where's it going?
Yeah, science, where we want this guy going.
Got
Um,
1, 2, and 3. Fine mesh open.
Roger
No,
that.
no, they're
Or do we want to put it in a bio box?
no. We can. Do you think it'd go through the slurp? It's pretty
Got
big.
slurp open.
It seems relatively flexible. Thinking we can do a little bit of a slurp on it.
The other
So
It's a little big, but I think it'll go.
the other option is to put it between the inserts in the bio box.
starboard bio box.
Sure.
There's a space in between.
There's two spaces and they're labeled A and B, so they're their own compartments. Go ahead and use them.
Okay,
Yes,
we will.
I see that on the sheets. Thank
So
you, Lisa.
starboard bio box.
Roger that. So starboard bio for you guys. All right, Jake. Roger that. You want to chase me over with the manip? Or yeah, thank you, with the bubble. Yes, please. Raj. Roger that. Go on ahead. I got some of my lateral going there. Hopefully it doesn't float away.
Yeah.
So are we going for the top part, starboard bio box?
We might have to disable the thruster with this
Yeah.
floaty guy.
Like this is gonna be a floaty sample.
Do we know if there's anything floaty?
No, nothing.
Okay.
It's about to be. It doesn't float away.
Boy, tough camera angle.
See if we can actually go to this one. I have a bit better visuals. Might be a more guaranteed sample if we slurp it, but we can try here. Oh, there goes my hand in the breadbasket there.
Caught in the cookie jar again.
Try to get it out without. Oh, there goes the sponge.
Yeah.
Slowly sinking.
Yeah.
Yeah.
Remind me to take out the wrist bias creep.
Roger.
Roger.
That's not very helpful right now. Doop-a-doo. Wow.
Well done.
Very good.
That is an
Okay.
interesting sample bio box there.
Yeah.
In between.
Let's not call it optimal.
Box coming in. Is that the
Roger.
forward or the aft part?
That is the forward.
Forward, okay.
That would be by E.
And that
And
sample number
confirming
is?
that was sample ID number NA124
Roger that, 021.
Dive salvo. Come back up.
Go ahead. Thank you.
And do you want to calibrate the wrist quick right now?
Yeah.
I'm going to do a, you're out of auto XY, so I'm going to do a reset.
Roger that.
Reset complete.
That was exceptionally well done, by the way.
Thanks, guys. That was good.
And Jake, you're a bit closer to the wall now. You can probably come up if you need to.
Roger.
Couple meters. But we're pretty stable.
20.
Right.
Back right there. On the other side of the curtain.
It looks better. All right, Rennie, you can go ahead and get started. I'll just finish stowing my stuff.
Roger that.
Oh, sorry, Science, did we still want to rock here?
Yeah. What do you think rock-wise, Greg, while we're here? Just take a look around.
We're full wide.
Roger that.
Not
Science,
bad.
you can give us instructions also on what we'll be doing.
What do you think? Size-wise. That look reasonable?
Do we have the lasers off?
No, lasers are off. Go. That might look reasonable. It's always, I think that might work. Yep.
More of a brown patina, so
Let's have a go at it.
give that guy a try.
Roger that. Change my wrist. Go ahead and push in there, Jonathan, please. That's great. Roger.
Hold position or do a,
Full wide, Jonathan.
yeah, I think hold position or transect or while we muster.
It's just out of reach, that.
Oh, okay, so you guys want to do another transect while we're mustering?
Yeah, scientists is going to be a little bit out of reach here.
Okay, I would say the problem with the transect is you can't stop and sample rocks if there's a good spot, and we need to get rocks before the dive ends, so.
I think you're muted, Greg.
Science, that rock's a bit too far out of reach. Did you still want me to move, get up and move? Or did you want to look for another rock?
Looking now, kind of hoping for another brown patina.
We could also, one potential option
That one,
is kind of going up here, upslope that way. Might be a few more, it's easier to park there. Up to you.
can you reach there?
That might also be a bit far. It's a little bit out of reach there, yeah.
Almost.
Close but no cigar for us.
It's a little harder to see. These ones are closer down here.
We're gonna suggest you guys move up the hill, so there's a hope of getting.
Okay,
Sure
fair
thing.
enough.
Sure thing. Maybe up by that rockfish there. There looks like there's some promising growth.
That's right.
Got it. Thank you. Lisa, we were going to suggest that they do a transect while we're out on the alarm.
That's a good idea.
So we can collect a rock here,
Yeah.
and then we'll be leaving in about 10 minutes on alarm, and they'll come. When we come back, we should be at a new place.
Okay, sounds good.
So Lisa, would you prefer we don't get a rock from here?
No, it's fine. If you can get one, go ahead and get one. It's a good use of our time because I forgot we're going to have a break here.
Right.
Jake, you want to look at the porch, please?
Yes.
Thank you.
Goodbye, sponge.
Anything around here look promising to you, Science? These might be good.
We're kind of trying to look for some with more irregular surface.
Mm.
Let's have a look at those.
Maybe up there. Arrows are better.
Yeah, they're a good size.
Roger that.
There's a 6-pack of them.
This might be a good spot here. The rockfish needs to move. So Science, would you like it kind of these guys here?
Yeah, can we have a zoom in, just have a bit of a look?
Sure thing. Jake, you want to look at the manip there, please? Go ahead, Jonathan.
We passed over the ones maybe that you originally looked at, Greg. Zoom out a little.
Come line.
That's too big.
Yeah, too big.
Finger.
Anything in this pocket up here?
Let's have a look at that.
Sure.
That looks like that's an okay size. That white sort of.
Go ahead and push in there a bit, please, Jonathan. Great. That one there?
Yeah. Let's see if we can pick that one up.
Sure thing. Pull away, Jonathan. All right, Jonathan, go ahead and fish in a bit, please.
Yeah, we are watching from the lounge. Sorry, we're not commenting. It's just they are all fine. Nothing is particularly exciting.
Can you tell what it is? Oh, it's very friable.
Very friable, yeah.
Okay.
Do you guys still want something friable like this?
No, thank you.
I think that was a no.
Now it's sand.
Roger that. Pull away, Jonathan.
So is that too big?
Yes.
Oh, sorry, move.
that?
That might be, let's see if that breaks if you try and pick that one up.
Sure thing. Jonathan, push in there, please. That's great. This is a nice size here. The bottom one here?
Yes, please.
Sure.
Got some tubes on it.
Great.
And if we can get a bit of a view of it before we box it. Kira, any advice on what this is? Oh, we've lost the spaghetti worm. So that's a terabellid worm. They're swimming up, teasing us with its presence.
It's like a fishing lure.
Hi, spaghetti worm.
It's in the
Oh, it's getting more into focus.
interesting
Goodbye.
maneuver.
We'll probably have its tube.
Yeah, that could be a phosphorite with a ferromanganese patina. It's hard to tell until we cut it open, but it looks pretty good.
All right, we'll box it.
Roger that. Full wide there, Jonathan.
Starboard.
Box number 4, green.
Roger that.
Rock fall.
This is going to be sample NA124 022.
022, roger.
Sample salvo?
Please go ahead. I'm going to look up at the manip there, please, a bit, Jake.
Yes.
That's rock number
Thanks.
4.
Green.
All right, go ahead, Jake.
Box coming out.
Rog.
Is it one of the back ones?
Yeah, so I can't really read which one's for. So is it the...
That's going to be starboard bio box on the starboard side of the starboard bio box.
Forward or
So
aft?
the forward.
Forward, yes.
forward, starboard?
And is it forward?
Yes, forward, I believe.
Forward and forward.
Forward, forward.
That's the back one.
What number is that?
I think it's that green color, right? In the front.
Yes, green color sounds
Sample
correct.
22,
4 green.
Okay,
4 green.
roger that. So this one here by where the sponge was?
Correct.
Yes.
Great. Okay. You
Great.
got it.
Thank you.
Fantastic.
All
Thank
right,
you.
box coming in.
Roger that.
We're probably going to be called away, so we think a transect might be best while we're out.
Sure.
Science, can you confirm what box you put rock 4 in?
That's gonna
Green 4.
in the starboard bio box, number 4. Green.
Coming back to dive salvo.
Thank you.
Roger that. All right, ready?
Okay. Let's just, it's a bit steeper, so let's just go at 0.2. If that's all right with you.
Roger that. Sounds good to me.
Go 225, and then once you pick up, we'll call it the start.
Rog.
Bridge nav. 100 meter step, bearing 225, speed is 0.2.
This is going to be transect number 5.
Roger that, transect 5, and it'll be
100 meters.
Yep.
And that will be sample number NA124023.